All right. We'll do the flower next.
And you said all these diagrams, like this list, is on Google Classroom or will be on Google?
It will be. It's not up there yet.
No. That is.
Hello,
Behind before.
You're gonna go back and watch this and be like,
Yes.
I'm gonna go back and watch.
You want to take it right now? Which reading quiz is this?
Jojo, are you recording right now?
She is.
Oh,
She is. I'm sorry. I probably should say that.
So we're
That one is. That one was. Yes, that was.
Or like, wait, it's
Carving
like
cycle.
one pen.
And. No. Oops.
All right. 2 more to go. You ready for the flower? Flower it is. So, the way I would start with the flower is I would do what is called the pistil first, which is all of the female stuff.
this is a die cut, right?
A dicot flower, yes. If they ask you for a monocot flower, the only real difference is the number of petals they can have. Monocots are
4 to 5.
3. I think
No,
they're
monocot are 3.
monocot are 3.
Dicot are
Dicots
4
are
to
4 to
5.
5. 4 or 5.
Remember that.
Multiples of?
That's
I
the one I
also
got
do.
wrong.
Because mono is 1 and 3 is less than 4.
Wait, say that one more time.
Monocot is 1 and 3 is less than 4.
So mono is closer to the number.
Yeah.
1 is closer to the number that they have, as opposed to dicots, which have a higher number.
Also, I remembered it by, like, we drew the monocot one first, mono was first, and dicot was second, and I just remembered that the first one had multiples of 3 and the second one.
So mono is 3 and dicots.
Yep.
4 or 5.
That's a difference between them structurally. Also that vascular tissue, also the number of cotyledons they have when they grow out. Anyway, so this is the female portion of the flower's system. Right next to it we can put in some anthers on filaments. And again, I'm not going to go crazy with these, but I'll put 3 on each side.
Do you just have all these diagrams, like, just totally memorized?
No.
Did you, like, look over it before this then? Or you
Yep.
just, like...
It's on my computer on the previous slide. But I need to maintain that air of, is it omniscience where you know everything? What is that?
Yeah.
Omniscient.
Yes.
Yeah, I need to appear omniscient for confidence. So that you guys actually believe the things I say.
Sorry, I blew your cover.
See,
No, I mean,
you
it's
have-
right there, so.
You and Mr. Anderson have the opposite approach with this. He, like, lets us know that he knows absolutely nothing.
Well, I know some things. To be clear, the reason I'm checking this is so that I don't mix up stigma, style, and stamen. You know, I know what a file is. Okay. That's what he says. Okay. So aside from and these are the male portions, and we'll label all this here in a second. We can go ahead and give this some petals, and if it's a dicot, you're going to want to do 4 or 5. So we can just do. And there's 1. Here's another one. Here's another one. Here's the last one. At the bottom, you have these little tiny, like, wimpy little leaves. We'll talk about those in just a second. And there you go. There's your
I just think about that every time I eat an apple now.
Right? That the apple is actually an upside down flower.
Yep.
Oh,
I think about it every time.
So if we were to label all of these, This is the ovary. Contains the ovum or the egg. This is the stigma, which is where it's the pollen landing site. The style, which is where the pollen tube would grow from where the pollen lands down to the ovary. Together, all three of those, this is not a great labeling job, but I'm just going to do this. All those together are the pistil. It's every day.
What is that?
It's just something in the ceiling.
Mysterious.
What is that mysterious? The anther is the portion that has pollen. This is the male part. It is supported by a filament. Together, those are called the stamen. Otherwise, we have. Come on, guys. Let's keep it together.
We can't. I can't.
Oh, fuck.
And just for kicks, we're going to label the stem too. Why not? Alright, I don't think I missed anything.
What's the sepals do again?
The sepals are tiny modified leaves that protect the flower before it blooms.
Oh yeah.
No, it stopped.
If you happen to drop 3 petals just to like on the diagram,
What Sarah just
would it be considered wrong, or because you're looking at like half of the flower, they would like assume that there was
Then it would be
petals?
6.
Well, so here's the picture that I gave you guys.
Huh.
Oh.
And
If it specifically says draw.
What if
diploid
it has
flower,
like
that would be wrong, right?
15 petals? Like if you find a flower in nature that has 15 petals, how do you know?
there's no way.
The leaves.
You could look at the leaves, you could look at the roots, you could look at the vascular tissue, but counting petals ain't gonna help.
You could take its seeds, replant it, and see how many cotyledons come up.
Or you could just look at the veins.
That is no fun.
Alright.
Oh man.
I was
They put as much
I
money
was-
as
Again,
possible.
on the-
Apparently when I was planting my beans and they came up with 2 flowers. I was like,
I was planting my beans.
for my IA.
Wait, I'm missing one. Where am I missing one?
And then I got really attached to them, so I found, but then they were so big that they started growing in circles. And so I found this like pot holder for like a planter. So I had like the holder as my things. And I flipped it upside down and ran twine all through it in different patterns. So
I
that it
remember
had,
you
like,
describing this.
twine thing so that it could grow through the twine.
Do you still have
And
this?
then, we lost power in our house while I was gone in Montana for like a week. We lost power and we lost heat and there was no heat and they all froze to death and died. And when it came back, they're all just dead. I was so sad.
That's what I'm talking about. I took mine from the school, and like I tried to cover them up so they didn't freeze, right?
Wait,
And I
Sarah.
got all their stems.
She's talking about her beans.
My bean plants.
It's like no.
Oh.
For my IA. It was after my IA. I just got really attached to them, so I replanted like 6 of them out of my like 25.
I thought you like.
If Joy was still recording, everyone.
And
Alright.
That's
It's
terrible.
Puppies that froze to death. Dave.
God,
Last-
it was horrible. Oh my god.
Last one. The human heart.
Oh, I don't need this.
Yeah.
Get it. I perfectly filled my paper. I'm
I,
very happy.
Hard.
I realized I don't have a human heart
Haha.
Do you still have
here
your
anywhere,
heart?
so I need to add it, but I'll add it later.
He doesn't have a heart.
I have no heart.
He's going in blind, guys.
Going in blind. Let's see what it looks like. Okay. I just drew the right atrium. The right atrium is connected by these atrioventricular valves. So give it like a little flap. The atrioventricular valve leads to the ventricle. These will then lead to another set of valves, the semilunar valves, which will lead out of the heart.
Looks like Mickey Mouse.
It's a question.
Similarly, over here, I'm going to do the
Yeah, if he
left atrium,
had it removed.
the left ventricle, and another set of, let's see, full valve, and then another valve.
Another.
Is it okay if those are overlapping?
And then
They can overlap a
Mine
little
are
bit.
really close to me.
They can overlap a little bit. I think if I was erasing or redrawing, I would actually want these to be a little bit closer.
So if we get a heart diagram on a test, we're like gonna have to like do it right the first time practically, because like there's a lot of stuff I'm gonna bring.
Just
That would be cruel. if we gave us
Erasable
a-
pen guys.
it's the hardest one.
Bless you.
Erasable
Thank
pen
me.
or test.
Okay,
I'm doing it for the bio.
Yeah,
So-
I'm doing it for bio.
let's
Well, no one else said bless you, so
now
I had
that
to
we
say it.
have
He would've
the
given
valves
you
and
I'm
the
sorry.
chambers,
I'm gonna give you that time.
gentlemen,
Sorry.
let's go and just walk our way through the entire path. You could enter the right atrium through either this upper area or the lower area. These are both the superior and the inferior vena cava.
What else can we call it?
The anterior and posterior
Yeah.
vena cava,
Yes.
I believe. And so both of those are going to lead into here. I'll put in lines later. I'm not sure if they would want you to draw flow lines, maybe only if they specifically asked for it. From there, the blood goes through the atrioventricular valves into the ventricle, through the semilunar valves, and then this is the part where it gets a little weird, because what we need to draw is basically a single tube that breaks into two tubes to go to both of your lungs. So up here, I'll have one, and then the other one we're going to ignore for a moment, but it's going to go that way. The problem is that right now what we need to think about is right here, there's going to be a tube that comes up in the front. It's called the aorta. We'll get there in a moment. Either way, we do want to close off these lines as we're doing this. So the blood goes to the lungs, gets oxygenated. It's going to come back. These are the pulmonary arteries. These are the pulmonary veins. And there's a set on both sides. Make those a little longer. And from the both sets of pulmonary veins, you enter the left atrium through your atrioventricular valves into the ventricle, through your semilunar valves, up and out this thing called the aorta. It's big. It swoops around the back.
Three little things.
And it comes out down there. And it does have those three little things, but I don't think you have to draw those. One thing we've sort of ignored to this point is like the outer perimeter of the heart. So we can go ahead and add some of those things in. I'll finish up my pulmonary artery right here. Give that one a line. And the reason that I'm waiting to do the outside of the heart is because it's the same reason why, actually, darn it, I want thicker cardiac muscle down there. My apologies. The reason that I would recommend waiting on this is for the same reason that it is easier to, if you're doing like speech bubbles, you write the speech and then you draw a bubble around it. It's tough to draw this stuff inside of a box, but once you make it, it's pretty easy to draw a box around it. So come up here. We'll meet the semilunar valves. Meet there, and meet there. Oh, that's a straight line because that's going around behind and like that. Okay. Not the most beautiful heart, but it's pretty good. And again, I would say that the most important thing is that you're keeping your lines as neat and connected as possible, and then what we can do is just label the crap out of this thing.
-1 for accuracy.
-1 accuracy.
So little.
Me too.
It hopefully encouraged you to make sure that you weren't inaccurate on your next diagram. That's the goal. That's the goal with feedback. Okay, so this is the right atrium, the right atrioventricular valves, the right, that's a V, ventricle. These are the pulmonary arteries. Arteries go away. These are the pulmonary veins. The left atrium. Left atrioventricular valves. Left ventricle. These are both the semi-lunar valves. And if you wanted to label them as right and left, you probably could. This is the aorta. Superior vena cava.
Is it COVA or CAVA?
Superior. This?
Yeah.
CAVA.
CAVA.
Vena cava. Cool. Lastly, I'll just throw in some quick flow diagrams. Blue is good for deoxygenated blood.
Did you get cardiac muscle?
Oh, I didn't label the thick cardiac muscle. No.
You're welcome. Sorry, that was sassy.
I deserve it. All right, oxygenated red blood comes in here, up and out, up and out. And of course,
Okay, that's in blue.
Haha.
there you go.
Funny.
Whew! Right? Nice job, everybody. All right. So, yes, Abe? Thank you.
Wait, should we say just cardiac muscle or should we say thick?
Not being able to release.
Thick is not a bad word, but you could just call it cardiac muscle.
Dayton's cardiac muscle.
Yeah.
Yeah, that's not the technical terms. All right,
Just pointing that
if you
way,
all could do me a
1970s.
favor, all of the stools need to go back up for the janitors so they can sweep floors and stuff like that.